Ladies and gentlemen, antibiotics are truly miracle drugs that have saved countless millions of lives. But antibiotic resistance is a critical public health issue that is eroding the effectiveness of antibiotics and may affect the health of each and every one of us. Ladies and gentlemen, my name is Panca Andana, and here I want to present you my presentation about my study that named Identification of Bacteriocin gene in Lactobacillus acidophilus. So we start from the introductions. Lactic acid bacteria, or LAB, have an ability to produce three kinds of antimicrobial substances. There are hydrogen peroxides, organic acids such as lactic acids, citric acid, etc. And then the last is bacteriocins. And Lactobacillus acidophilus is one of lactic acid bacteria that known have a possibility to produce these three kinds of antimicrobial substances. And then Lactobacillus acidophilus from American type culture collection, or ATCC, that grown in America. And then Food and Nutrition culture collection, or Panca 001, it's one of them. So it have a possibility to produce this kind of antimicrobial substances. And especially in Food and Nutrition culture collection, or FNCC, there are no references yet that this Lactobacillus acidophilus from FNCC have a possibility or have an ability to produce antimicrobial substances or produce bacteriocins. And then bacteriocins is so popular and among the researchers. There are so many references that talking about bacteriocins. Why? Because bacteriocins has certified from CPS from US, and then from has a GRAS or certified from US FDA, and then it has a broad spectrum. So it meant is effective in gram-positive and gram-negative bacteria, and also even it's effective in multi-drug resistant bacteria. So it have an ability to stop the growth or kill the multi-drug resistant bacteria. And then it has thermal and pH stability even in extreme temperature and extreme pH, and then it's low toxicity. Of course, it is important because the low toxicity means man can consume it and then have an antibiotic effect. Meanwhile, it's from the natural vine, right? And then the production of bacteriocins depend on its strain in culture optimization. So it means other strain in same species has a different name or has a different bacteriocin that they produce. And then the genes encoding bacteriocins are located in chromosomes, so here, or even in plasmids. And then the aim of this study is to detect the bacteriocin gene in Lactobacillus acidophilus Panca 0051 and in ATCC 456. So the method, the bacteria that we test is the bacteria that we said before, Lactobacillus acidophilus ATCC 456 and Panca 0051. And then the negative control is the non-lactic acid bacteria, Bifidobacterium vividum, aqueous or distilled water, and then nucleoside water. And then the primer that we use is the specific primer for acidocin LF221A. So it's the one of the type of acidocin, because there's so many bacteriocin name that have found. And then this is the primer, the specific primer that we use, the forward and the reverse primer. So the first thing first, we sterilize the utensils and the tools that we use for the study, and then after that we continue by bacterial growing in agar plate or bacterial rejuvenation, and after that we made the bacterial suspension. So we made three bacterial suspension there: Lactobacillus acidophilus ATCC, FNCC, and Bifidobacterium bifidum. And then after that we run the DNA extraction by Agilent DNA extraction kit by Agilent Technologies. So first thing first, we centrifuge the the suspension to lysis the cell. So we separate the DNA and then the membrane plasma from the cells. So it separates the DNA and other organelles. And then after that, we add the buffer. So and incubate it. The buffer adding here, the functions is to lysis the amino acids and protein like enzymes, RNase and DNase and so on. And then the RNA itself, we lysis it. So basically, we lysis all organelles except the DNA. So the DNA is pure. After that, we purify it. The DNA that we have extracted, we wash it by pure ethanol or 100 ethanol. And then after that, we storage it. We save it in a low temperature and in buffer. And then after that, we run the PCR. So the PCR is basically just the PCR or the thermal cycle will denaturate the double-stranded DNA, and after that, after denaturation, we have the annealing process. So it means the polymerase, and then the specific primer will bind to the DNA target, and after that, we go to the extension or elongation process. So it means we made the new DNA. So the thermal cycle basically will copy this DNA target more and more. If this process which I saw in the video is just one cycle, the PCR will go around 35 and so on cycles. So it means so many amplifications from the DNA target. So the amplicon or the PCR products that contain the DNA target that we have will go to gel electrophoresis analysis. So we have to know the size of the DNA that we amplify in the PCR. So this is the result. From the gel electrophoresis, this is the first lane is the marker. So it means the first band here is have a 10,000 base pair, and so on. And then there are the first lane, the sample lane is the first lane that from Lactobacillus acidophilus FNCC from the first DNA extraction, and then Lactobacillus acidophilus FNCC from the second extraction. So we run two then the extraction, and then ATCC 45.6, Bifidobacterium bifidum, distilled water, and nucleus-free water. So here the Lactobacillus results show that there are band in 100 base pairs. So we run the literature review or references say that the gene for acidosin electron 221A or a primer that we use has a gene size or DNA size around 100 base pairs, so it matched the references. So it means the lane 2 and lane 3 has matched the primer and match the references that say that the DNA for acidosin F221A has 100 base pair. So it means the Lactobacillus acidophilus FNCC 0051 and ATCC 45.6 have a gene encoding bacteriocin, and then Bacterium bifidum doesn't have gene encoding bacteriocin because it's non-lactic acid bacteria, right? And then not all the extractions succeed. So it show in first lane that there are no band at all because the DNA extraction from the first attempt is not succeeded. And then the negative control, so that there are no contamination at all in the process. It means why? Because there are no band at all, in the lane 3 to lane 6. So the negative control has no show the DNA in a 100 base pair size. So the conclusion, Lactobacillus acidophilus has an ability to produce bacteriocin, but none on non-lactic acid bacteria like Bifidobacterium bifidum. Well, thank you very much. Goodbye.